Hello everyone and welcome to today's live broadcast, Open Targets Platform, Mining Gene and Disease Evidence for Improved Drug Target Selection, presented by Dr. Denise Carvalho-Silva, Scientific Outreach Officer, Open Targets, EMBL-EBI. I'm Julie Simmeroff of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. For questions not answered today, we will follow up with participants via email. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the icon located on the lower right of the screen. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now, please join me in welcoming Dr. Carvalho-Silva. I will now turn the presentation over to her.
Okay, so good morning, everyone. Thank you, Julie, for the introduction. I hope you all have been enjoying this virtual conference, and I'm very pleased to be part of it. So thank you very much again for the organizers for reaching out, contacting Open Targets, and inviting us to take part of this great event. So as Julie said, I'm going to walk you guys through Open Targets. So we'll be doing this for the next 45 minutes or so. And so my name is Denise, and I've been working with Open Targets for a year now. And we are based here in Kingston, just outside Cambridge in the UK. So what I will cover in this talk, I will tell you guys what Open Targets is, what the consortium is, what we are all about. Then I will tell you the experimental projects that we are involved with and spend a little bit more time going through the platform. So it's a web interface where you can mine genes and disease associations. Towards the end, I'll try to highlight what makes Open Targets unique. And then I will also give you some idea what our plans for the future. So Open Targets is a consortium with more than 200 people involved with, mainly in the UK and also in the US. So the talk that I'm presenting here today is by no means my own work. So this is the contribution and the hard work of many people within the consortium. So the credits should go all to them. So some of you may be involved in the field of drug discovery, whereas perhaps others of you, you may be completely new to the field of drug discovery. So you may not be aware of how long it takes to identify a drug target, to produce that drug, and then to have it sold by our pharmacist. So it's a very lengthy process. It can take up to 20 years. So from the very start, so you have an idea, so you start doing some identification of your drug target, perhaps you start doing some research, basic research. You will probably be using some model organisms, such as mouse or zebrafish, in order to test how safe your drug is in those models. Then you can go on into the clinical trials, which usually are the most expensive part of this drug discovery path. The clinical trials are divided up according to different phases, and eventually, if all goes well, your drug will be approved by the FDA, for example, and you'll be able to provide that to the members of the public. So there are many challenges in the field of drug discovery. So despite being such a lengthy process, it is very costly, especially those stages of the clinical trials when you're dealing with healthy volunteers or you're dealing with patients. And unfortunately, the success rate of discovering new drug targets is very, very low. And it varies depending on the therapeutic area that you are working on. So for example, within cardiovascular diseases, it's where we have a higher percentage of success. So it's around 20%. So this means that there is a high attrition rate. You can think of attrition rate as the number of dropouts. So the number of drug compounds that you start with in your journey of drug discovery compared to the number that you end up with. So the number is very, very, very low. And the causes for this dropout, so the causes for the attrition can be several-fold, and also they vary depending on the phase or the stage of the drug discovery timeline. So for example, when you are working on the preclinical phase, which is when you are using model organisms, you're using mouse and zebrafish. So the main reason for the attrition there is the safety. So 50% of drug targets, they tend to drop out because they are simply not safe. So this changes along the clinical phases or the clinical trial phases. So whether you move on to phase 1, 2, or 3, the main reason for the dropouts is efficacy. So simply the drugs or the drug target that you are working on and doing your research, that drug target is simply, it simply doesn't work. So there are different causes for this attrition. So once upon a time here in Cambridge, in the UK, we had those 3 people having lunch, and they were just talking about drug discovery, and they realized that probably there is room for improvement here. So could we improve the way that identification of drug targets is done? Can we improve it? So yes, we can, and yes, we should. And it was also clear for them that one institution, one lab could not do this on its own. So those people were the director of the Sanger Institute here in the UK. We also had, at this lunch occasion, we had Patrick Valens, which is the president of R&D in GSK, and we had also Professor Janet Thornton, who at the time was the director of EMBL-EBI. So those three minds got together and launched this idea of open targets. So they launched the consortium. And our main vision is to transform the way that drug discovery is done. So we would like to combine genomic experiments. We would like to include rigorous statistical analysis and also computational techniques to help identifying drug targets and also to allow for the association between drugs, genes, diseases, and pathways. The major core principles of the consortium is that we are pre-competitive. So this means that we work very much in the early stages of the drug discovery. We are also aiming at publishing our data as soon as we have got hold of them. And we are also open to new people joining us, or new groups or new institutions. So those three minds that I talked about earlier, so those are the Sanger, GSK, and API, so they were the founders of this consortium. So they were our three founding partners. And the consortium itself was launched in March 2014. So it's just over 3 years old now. And we've got a very bold objective. So we would like to be the world leader for human target discovery. So who is Open Targets now? So although we were founded by GSK, EMBL-EBI, and the Wellcome Trust Sanger Institute, more recently we had Biogen joining in. So Biogen is the oldest biotechnology company in the world, and it's based in the US. Its headquarters are in Boston, and they are very much interested in diseases such as Alzheimer's, Parkinson's, and they have joined Open Targets in 2016. So now those are the four major institutions that make up Open Targets. But as I said, this is not an exclusive club. If you want to join Open Targets, just get in touch with us and we can move from there. So this is a little bit of the background. So now I would just like to highlight the major areas of research within Open Targets. So we've got two major umbrellas. On one hand, we've got people working mainly here at the Sanger, but also at the GSK, working in the labs. So people creating new data. They are working on single-cell RNA sequencing. They are using organoids. They are using organoids as well as models of disease, and very much using CRISPR-Cas9 gene targeting as well, and gene editing. So they're very much using state-of-art and very high throughput technologies as well to create new data using human cells or organoids. So this is one of the two major projects within Open Targets. So it's creating new data that are at the moment completely unheard of. On the other side of the coin, we also have the bioinformatics aspect of Open Targets. So at the moment, what we are doing, we have integrated data that are publicly available. We've integrated that into a platform, a web interface, so that people all over the world can go to that place and mine genes and disease associations. So you can do so by using our website, but if you are programmatically inclined, if you know Python, for example, or R, you can get hold of our data through our API, or also perhaps through the data dumps. So those two are the two major umbrellas within Open Targets, and they are concurrent. So they are happening very much at the same time. And if you want to know more about our projects, I will strongly recommend you guys to have a look at our link over there. So opentargets.org/projects. So from now on, I will focus on the platform itself. Before I do so, I would just like to perhaps highlight that within the experimental project, we are mainly focusing on oncology, so different types of cancers. We are also very much working in the field of immunology, and also working with Alzheimer's and Parkinson's disease. So those are our major focus in terms of diseases when it comes to create new data. So the Open Targets platform, which is this other side of the consortium, so it's a web interface which was developed by my colleagues here at the core bioinformatics team at the EBI. And we would like to allow scientists to use this platform in order to identify target and disease associations. So by target here, I mean a gene or a protein. So the platform is very much frequently updated, and in each of those updates, we may have new data, and we can also include new web features or new tools. So everything that would allow you to identify gene and disease associations. And one of the most important aspects of the platform is that everything that we do here is very much driven by you guys over there. So we take into account feedback from our user communities, and before implementing a new web feature or a new tool, we always consult with our user communities to see whether or not this would tailor your needs. So this is the URL for the Open Targets platform. So it's simply www.targetvalidation.org. Although the consortium itself was launched and founded in March 2014, the platform was released for the very first time in December 2015. So it's very much still evolving. So it's just over a year old. So it's very much new, but it's moving and it's improving as we speak. And one of the most interesting aspects of the platform as well is that it contains very funky and appealing visualizations. So it can catch your eye very easily. So this is our model in order to make associations between targets and diseases. So it's important to try to keep this model in mind. So in order to make the association between targets and diseases, we need evidence. So we need pieces of evidence that can support that association. And at the moment, where do we get these pieces of evidence from? So we get them from publicly available databases. So on this table over here, I'm just listing the current set of data that we've got in the Open Targets platform. So when it comes to genetic variants or mutations, so we get those from places such as the GWAS catalog or UniProt, and also EVA, which is the European counterpart of DB-SNP, with the added value of having clean VAR data. So we've got information of clinical significance. When it comes to mutations that cause cancer, so we've got those as well, and they come mainly from the cancer gene census. We also have got data that shows whether or not a gene is differentially expressed in healthy and diseased cells. We've got drug information coming from Campbell. We've got pathway information coming from Reactome, and we also mine the literature. So we mine millions and millions of scientific papers out there and research articles, trying to find the link between a target and a disease. And we also have data on mouse models. So all of those disparate data sets, they've been combined, integrated, and used so that we can give you millions and millions of associations between thousands and thousands of genes and thousands of diseases. This is by no means a comprehensive list of data sets, and we are very much interested in hearing from you if you've got a favorite data set that you would like to see in the platform. So if that's the case, simply get in touch and let us know. And one of the bedrocks, perhaps, of the platform is that the diseases, which are around 9,000 of them at the moment, human diseases, they need to be classified, followed an ontology. So you can think of an ontology as a dictionary that provides the relationships between diseases. So here, for example, we may be looking at asthma. There we are. So we know that asthma is a child disease when compared to respiratory system disease. And we know that asthma has got a subtype, which is childhood onset asthma. So this is what we call an ontology, a disease ontology. So we can get the relationships between those diseases, between those entities. So the ontology that we rely on is EFO, which is the Experimental Factor Ontology, created and maintained here at the EBI. So EFO provides you with this controlled vocabulary. So you can either search for example, Alzheimer's without or with the apostrophe, and you will get the same results. And we also provide the hierarchy. So you can see what's the relationship between your disease of interest and its parent and its child terms. So there are several advantages of relying on an ontology. And we very much need consistency and easier ways to automate this whole process. So this is just to give you an idea how the data flows in our pipeline. So don't worry too much about the details here. It's just to give you an idea, again, of the information we've got in terms of diseases. So we've got data coming from Orphanet for rare diseases, diseases that have got, Mendelian inheritance. We also have data for common diseases such as, diabetes, some types of diabetes, perhaps some types of cancer as well. So we try to be very comprehensive with the platform and include as many diseases as we want. We get our SNP data from EVA, and we validate all the data from those data sources. We make the association between a target and disease. So remember that data model of ours. So we make the association and we look at, and we give and compute a score to give you an idea of confidence, how confident you can be on that association. And you can also get data on the target itself, on the gene itself, and also you can get data on the disease. So it's a profile of the disease or a profile of the target. So I will skip this. This is just to show you how the data looks behind the scenes, and also what are the factors that we take into account, such as this one highlighted in red, and also the final association score here for a given data type. So again, just to give you a flavor of the sort of data that we hold and we handle actually behind the scenes. So regarding the score, so yes, so again, this is our data model here. So we use different types of data sets to make the association between the target and disease. But we need to give you some sort of confidence on that association. And we do so by giving you a score. So again, I won't have time to go through this, but this scoring is a statistical approach that was developed by my colleagues. You can get 4 different types of scores. You can get one at the evidence level, at the data sources level, data types level, or the overall score here. So you can get hold of those numbers and make your own judgment call. The score will vary from 0 to 1. The closer to 1, the stronger we believe in that association between a target and a disease. So this is just a summary of the whole process there. So we've got our Open Targets database here. We've got data sets coming from external databases. Those are all publicly available. They have been combined together. They have been scored. They have been quality checked. And as we speak, we've got our colleagues at the Wellcome Trust Sanger Institute creating new pieces of evidence for oncology, immunology, and also neurodegeneration diseases such as Alzheimer's and Parkinson's. So as soon as this data is available, it will be fed back into the platform, or it will be fed back into the public available databases out there. And here I just have a screenshot of how you can see this data in the platform. So one of the things we provide is this heat map with different shades of blue. The darker the blue, the stronger is the association between your gene of interest and a given disease. So we do support decision-making when it comes to drug discovery and drug identification. So we can tell you which targets are associated with a disease. You can use the platform to find out about the mechanisms of this disease of your own interest. You can use the platform to find out whether or not there are drugs that are FDA approved for clinical trials or drugs that are already on the market. And you can find lots and lots of different types of data for your drug target of interest, your gene of interest, and many, many, many more other use cases. So this is just 4 examples of things that you can do with the platform. So you can start from the disease point of view. So you can search in the platform for your disease of interest, let's say multiple sclerosis. So again, here, just the reminder for our URL, so targetvalidation.org. So you go in there, you type any disease that you are interested in, and you can get a list of all possible genes associated with that disease. So this is how it would look like. We've got this table, which is a heat map. Again, different shades of blue over there. The darker the blue, the stronger the association. So the very first column is the target name or the target symbol. So it's a gene symbol there. Then you've got the overall association score. And the following columns, the 7 following columns, those are the 7 different data types. So we've got genetic associations, somatic mutations, drugs, pathways that are affected in diseases. We've got data on expression of RNA, which varies between disease and healthy tissues. We have data being mined from the literature, and we also have got data coming from mouse models. So for each of those 7 data types, we've got a unique association score. Each of those columns can be sorted. You can use this search box there in order to filter the table. On the left-hand side here, you can filter by the data types, where there's genetic associations or drugs. You can also filter by pathways. You can filter by the class of your target, for example, whether they are membrane receptors or enzymes. Or perhaps you can filter by including your own list of targets within multiple sclerosis here. So there's a lot that you can do. So please go ahead and have a look at this. Have a look at our have a look at our platform and give it a go. So one of the things that you can do as well, so if you click on this link here called View Disease Profile, you will be taken to a page that looks like this. So the disease profile page, which is here, this URL. So this page will give you information such as the phenotypes associated with multiple sclerosis. It will also give you all drugs in clinical trials targeting this disease or using patients suffering from multiple sclerosis. And you can also get the disease classification, which is the ontology. So you can see that here on the right-hand side. In green is my disease of interest, multiple sclerosis, and in blue you've got the different subtypes of multiple sclerosis, and in red you've got the parent terms within the ontology of multiple sclerosis. And here is a table just listing all the 32 unique drugs currently in clinical trials with patients suffering from multiple sclerosis. So that was the disease-centric workflow, but you can also use the platform and start your journey using a target symbol or a target name. So if you're interested in a given gene name, just go to the targetvalidation.org here, type in your gene name, let's say BRCA2, and then you can find all the diseases associated with that target. You will find then breast cancer and many other types of diseases associated with BRCA2 in this example. And you can drill down and find all the possible pieces of evidence that we in Open Targets have used to make the association between BRCA2 and all the diseases that we display in our platform. And finally, so far I have given some examples on how you can perform this search using the platform, and it's usually on a one-by-one case basis. So you can type in BRCA2 and you can get the list of diseases, and then you can type in multiple sclerosis and you get the list of targets, but perhaps you are already working on a list of your own favorite targets. So let's suppose that you've got some experiments that highlighted up to 200 genes or targets that you would like to pursue further downstream. So you can, ideally, you would like to perform this search in a batch way. So you would like to search for all of them in one go. So very recently, we released what it's called the batch search tool. So it's www.targetvalidation/batch-search. So over there, you can simply either paste in your list of targets, or you can upload from your desktop, and you can run that tool, and we will show you every single piece of information in terms of all the diseases associated with those hundreds of targets of yours. We'll show the drugs for those targets. We show the pathways those targets are involved with, and possible interactions between those targets in your list. So these are very much ways of accessing the data via the web interface, and you will have the funky visualizations, and it's very intuitive and very easy to use, and it's perfect if you're working with 1 or 2 targets, or perhaps with 200 targets, but not more than that. So there are alternative ways that you can get hold of our data. So you can download the entire data sets in Open Targets. Just be aware that the size of those files are simply huge, but nevertheless, we do make that available to you, so you can just download those and start mining the information that you want to on your terminal window, for example. So there won't be any visualizations there for you, but the data, you can just do whatever you want with it. If you rather have extracts of our data sets, so this is in between the web interface and the file data dump, so we can do so using our API. So if you've got some knowledge of computer programming, it's very easy to use, and we have a REST API. So again, I won't have time to go through this or give more details, but I think it suffices to say and to highlight that there are alternative ways to get hold of the data, and we would like to encompass everyone in the scientific community, those who know how to program, but also those who do not know how to program. So whichever group you belong to, you will have a way to get hold of our data. So I think we've got perhaps less than 20 minutes left between. So I would like maybe perhaps just try to wrap up by highlighting the reasons why we think Open Targets is unique. Why is this consortium unique? So first of all, we are trying to address as many areas of human diseases as we can, especially when it comes to the platform. We have no bias whatsoever. Providing the data is out there, publicly available, we will integrate the data and provide it to you. We also very much put our users first. So we want to hear from you guys what is missing in the platform, what we do well, what we should improve on. So there is no point in providing this platform or providing this data and them not being useful to you guys. So you guys need to come first, and you do come first in this consortium. We also do work at the genome-wide level, so we are interested in protein coding genes, in non-coding genes as well, intergenic regions, and we are looking throughout the human genome as well. So we don't focus just on exonic regions, for example. And we perform genomic experiments, but also transcriptomics, and we'll be doing some metabolomics research as well. And another very unique aspect of the consortium is that we do bring the partners together. So at the moment, those are our 4 current partners. So Biogen there in Boston, we've got EBI here in the UK, GSK, UK and US, and also the Sanger Institute here in the UK. So those 4 renowned institutions working together, being brought together by the consortium, exchanging information, exchanging ideas, collaborating together, sitting next to each other. I've got a colleague from the US sitting here next to me in the UK. So we are very much collaborative, and everyone has got a say within the team. So what is coming up next? So at the moment, you can search for a target name or target symbol. You can also search for your disease of interest. You can search for drugs. So you can type in a drug, whether it's a common name or a commercial name. You can type that in, and you can get the information that Open Targets has integrated for you. You can also search for phenotypes. So what is coming up next? We would like to allow you guys to search for genetic variants, such as RS numbers from DB-SNP. We would also like to allow you to search for pathway IDs and also PubMed IDs. So the other thing we would like to provide, at the moment, we provide only target and disease associations, and we are working on visual ways to represent target and target associations and disease associations. So the scope or the universe of associations will go beyond target and disease only. And the other thing we are doing at the moment, we are taking into account functional genomics and functional studies in order to assign genetic variants to drug targets. So that will be very much, perhaps by July, by September, you will have ways that you can assess some functional information as well that will be included in the platform. So I would like just to mention that for more details, I would really recommend you guys to look at this publication here at the on nucleic acid research. So we are very pleased that this paper of ours was selected as a breakthrough article in the end of last year. So it's a great place to go and get all the detailed information on how the platform was put together here at the EBI. And finally, I would just like also to say that we are very much open to hear your comments, your suggestions. We do have an outreach team here, so we give you support. So the best way perhaps to get in touch with us is by emailing us, so you can use this email address there. But we are also very much active on social media, giving you hints and tips on how to use the platform. We are every now and again, we are recruiting new members for the team. So it's a good place, especially our Twitter account is a good place to go and have a look at the jobs that are available there. So, and we've got lots, actually we've got 2 GIF animations as well, which I highlighted there for you. So you can go to those, sit back, relax, and for a minute or 2, you will see how you can navigate through the platform. I won't have time to do a live demo here, but you can just check those GIF animations and they will give you an extra flavor of what you can do with the platform on a one-by-one case basis. And here I've got an animation on the batch search tool, so you can see how you can search for hundreds and hundreds of targets in one go. So I think that's very much what I wanted to say. So I hope it was useful for you guys. So I think now perhaps we may be open to a couple of questions. I'll be delighted to answer questions if there are any of those. Thank you very much.
Thank you, Dr. Carvalho-Silva, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, we will follow up with you via email after the presentation. Let's get started. Our first question is, how long does the platform get updated with new data?
Okay, yeah, thank you for your question. Yeah, so the platform at the moment, it gets updated every 2 to 3 months. So the pipeline is rather time consuming as it stands, but we are working on trying to speed that up a little bit. We are aiming at providing updates on a monthly basis, but as it stands, it's between 2 to 3 months.
Great, thank you for that answer. The next question is, do you have information on drugs which clinical trials have failed?
So our information on drugs come from Campbell, which is a database for drugs and compounds here at EMBL-EBI. So at the moment, we don't have a way to find out whether or not the drug failed on clinical trials, but we are working on ways to incorporate that information. But at the moment, we don't have information on failed drugs.
Appreciate that. And we'll wrap up with one more question. And that question is, do you have proteomics data?
So when you search for a target, so let's suppose BRCA2, which is the gene or target associated with breast cancer. So when you search for BRCA2, so you can get proteomics data coming from the Human Protein Atlas database. So you can find out what is the expression level at the protein for that given gene, and we are now working on trying to incorporate more proteomics data and also give a more comprehensive landscape for your target of interest.
Great, thank you so much. And that will do it for the questions for today. I would like to once again thank Dr. Carvalho-Silva for her presentation, and I would like to also thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 25th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.